discuss cell biology with numbers. I will focus on size. On the first chapter in the book of Milo and Phillips. So first I have some slides that I'd like to share with you. So this is just to give you an idea about the topics I will discuss today. I was telling you the other day there are some paradigmatic case studies in cells, in humans, in bacteria, in yeast. And today I give you at least here some images that I took from the internet, from different experiments. So these are human cells. As I told you in the first lecture, they are not in general spherical. This is the way they look. This is a real image from the microscope of a human cell. So nothing else like a football, but something extended that moves, that grows, that divides. You can see it in purple, the nucleus. You can see the cytoskeleton, blue, and this
so we can't see the screen like that. Can you share it?
You can't see? Okay.
Yeah, share the screen,
yes.
please.
Okay. Do you see it now?
No. You have to click
Yes,
the
I see now.
Oh,
You see it now, but I don't see it. So. Okay.
Function F5.
Yeah, now, do you see it? Idris, you see it? Okay, very nice. And I think I'm in slides only. I was showing your colleagues here in Trieste an image of human cells. You see the nucleus. This is from the microscope. The nucleus is in purple. The cytoskeleton, it's in blue. And also this laminopodia, which is like so part of the cytoskeleton, are in red. So these cells are dynamic objects, so they move, they divide, they grow. So you don't think about them as a static object. There are different types of cells. And here are some simple examples. Red blood cells, what we have in the blood. Neurons, which have, you see the red blood cells look like discs. The neurons look like fibers, long fibers. And the last ones are some that I have been investigating. These are sensory cells. They are in the cochlea. And you see they have very large body and some protrusions. This is called the hair bundle. I will talk about this during the course. These were human cells. I'll show you now bacteria. So this is what I was trying to show you the other day. They have this type of cylindrical shape, E. coli. And if you look at a needle before, on the left, and after the use, you can see how bacteria are colonizing the tip of the needle. So this is to give you the idea of what is the size of a bacteria. So you take a needle, you take a microscope. In the microscope you can see objects of the size of a micron. So you have them there. And plant cells, I have an illustration I found on the internet. So they are a bit different. They form this type of mosaic. They're more robust, let's say, more square shape. And they have this membrane that covers on the matrix connecting the cells. They are typically green because they have organelles called chloroplasts that I will talk about. And finally the virus, which I will talk a little bit today. And these are two examples, classical examples. One is the Bacteriophage, which is, you see on the top, there are the green guys trying to kill a bacteria. They attach to the bacteria, they put the DNA inside, and then they create new copies of them. And in the bottom is an old image in the microscope from HIV. You can see here the membrane and you see the spike proteins are common with the coronavirus that I will discuss later. Well, as I said, these are things that move and have dynamics. And this is a very nice movie of a cell division. You see the cell is dividing into daughter cells. And the green are what is called the microtubules, that I will also talk about them today. And the red are the chromosomes. So this is the genetic information in the cell that is condensed in these fibers and it's duplicated during mitosis. So it's a very beautiful movie. You can see many nowadays on the internet, and there's a lot of research about the forces, the velocities, and so on of this process. So I think with this, I'll stop. Later I will talk to you a little bit about coronavirus. So have something to tell you about this. Let me go back to. Okay, so this was the introduction. I hope it's good for you. So today I'll discuss about size. So let me move this up. And I have to switch it off.
Yeah.
Can you tell me where is the button? Okay. Oh, here. Very good. And the door. You can close it. All right. So first of all, I will put a scale bar here, which will be the scale of size in meters of different elements in biology. I put here 10 to the minus 9 meters, 10 to the minus 8. Things are small, typically. 10 to the minus 7, 10 to the minus 6, 10 to the minus 5. Okay, so the first thing I will ask you, this is called the nanometer, this is called the micron, and so on. The first thing, as a reminder, is what is the size of a water molecule? Do you remember?
What?
A water molecule, molecule of water. Here? Well, imagine an atom. How big is an atom? You remember? One atom. It's 1 micron, 1 atom. Typically, an atom is 1 angstrom.
1
No?
micron.
1 angstrom. 10 to the minus 10.
Yeah.
Water molecule is in this here. 1 hydrogen atom has the size of 1 angstrom, 10 to the minus 10. Water molecule is here. The protein is a bit bigger. You can put it around here. The protein is of the size of nanometers. These are nanometers. Protein is of the size of nano. A bigger unit is the ribosome, which can be in this size, a ribosome. This is the machine that produces the proteins from RNA. Also, a virus. There is a virus. Where do you put the virus? More or less. 10 minus 6, yeah, microscopic, yeah, somehow. Here, between 10 minus 7, 10 minus 6, for example, is HIV virus. And a bacteria. Is it here a bacteria? What was the size? On the right, no? A micron, microscopic. We can put E. coli, for example, here. Coli. And a human cell is bigger, as I said. This is a mammalian cell, not only human. Cell is here. So this is the typical sizes, and I will discuss most of them and tell you about their sizes and characteristic sizes. So I'll first start with cells and viruses. Cells and viruses actually will be here. This box will be cells and viruses. But I will also discuss.
Shouldn't HIV be in that box as well?
Yeah, exactly. This is good. You are here. Then there's another box which is for the organelles of the cells. So the organelles are somehow like this, more or less in this size are the organelles. Like a mitochondria, chloroplast, etc. And then these are the molecular components, like for example glucose, the lipids, etc., which, following the notation of the book, they call it nuts and bolts. Okay. But these are mainly the molecular components, the molecules. Water is one type of molecule. We have another one, for example, the nucleotide is a molecule. The lipid is a molecule that is in the membranes, etc. They are here in this box. I start from the big ones, cells and viruses, giving you an appetizer about their size. So first, I discuss the viruses here. So with a sketch, it could be following. So we have this guy, we have his structure, and then has this type of shape. This is the virus infecting a cell. This is the membrane of the cell. Here is the virus. And this one that I'm putting is the one that's called this, Bacteriophage. This means in eating, and this means bacteria, so these are killer of bacteria. And this virus has RNA inside. This is RNA. This part outside is called the capsid. It's made mainly of proteins and lipids. Capsid. This is called C. This is like the structure from where the virus introduces the RNA in the cell. And what is important is its typical size. This size here. This type of virus is of the order of 100 nanometers. It is not as big as a bacteria. It is 1/10 more or less as an E. coli bacteria. The typical image of what this virus does is there is an E. coli, an E. coli bacterial cell, and this virus comes here, bacteria. They are able to bring the DNA inside the bacteria, and this DNA is used by the machinery of the bacteria to produce proteins. Later on, this produces that this bacteria, these viruses could be destroyed, for example, but inside this DNA gave rise after transcription to other viruses which have a copy of the RNA of the virus. And these guys then proliferate and break the membrane of the bacteria. So they ultimately kill the bacteria. Okay. This is just to give you an idea. An interesting number is how many viruses are on planet Earth, roughly. So this would be, what do you think, more than humans or less?
Definitely more.
Definitely more. So there are around 10 to the 31 virus in the biosphere. And now a question is, how much is this in comparison to how many humans? So we have 10 to the 31 virus. And how many humans are on Earth? 1 million? No, more. 10 to the? You don't know. 10 to the 9. Around 10 to the 9 humans. 1 billion in some of 10 to the 9. And every human has around 10 to the 13 cells. So this means that there are around 31 divided by 22. This is around 10 to the 9 virus per human cell. So on Earth, per human cell, there are 1,000 million of viruses. This is very scary, you could think about. However,
But not all of them infected.
no, of course, we are not going to encounter all of them, fortunately, no? But this is about the numbers. And another thing is, what is the mass? These are very light objects. These are very small, tiny. So even though there are many, they are most populated species on Earth, they are very light. So they are only 5% of the biomass. Okay. And of course there are many types. I was telling you about this one. You can have, this is a virus with DNA, but another virus very different is HIV. Well, I'm doing a very bad illustration. HIV has lipid bilayer recovered by proteins. These are like spike proteins like in the coronavirus. And inside has, okay, sorry, here I made a mistake. Because this is DNA. Okay, this is DNA here. This is standard virus. But HIV doesn't have DNA, but has RNA. And it can later on transform this RNA into DNA. So this is a little bit of different type. Just to give you an idea of what type of machines are the virus. Right, so this is the size of the virus. About bacteria, is another type of cell which I already discussed very much. For bacteria, I focused on E. coli. So in E. coli, remember, these are the flagella. Of course, I put always the flagella here, but you have to think about the flagella is here, and the flagella can rotate, and so on. It's not that there are two legs attached. No, they can have many. And this, they have motors here that enable the bacteria to swim and rotate in 3 dimensions. I'll talk a little bit about these motors later. So it's a much more complicated thing than what I'm drawing. These are the proteins, the ribosome, etc. This is the DNA. And as I said, well, this is the cell wall. There is a membrane here. Cell wall. And as I said, the typical size is of a micron. So the volume is around 1 micron cubed. Remember, this is 1 femtoliter. And we can also think about what is the mass of this object. The mass is, what is the mass? How can I calculate the mass? You know the volume?
Density?
Oh, this should be fast. Density?
Yeah. Mass into volume, so mass.
Density is mass divided by volume.
Yeah, mass over volume.
Okay, very simple, eh? Density is mass divided by volume, so mass is rho v. Okay? Density times volume. What is the volume? So the volume is 10 to the minus 15 liters. And what is the density?
We did that.
Let's say it's made of water.
Then 1 kg.
1 kg per.
Meter.
We have here liter. So
Yeah,
1 liter is 1 kg, no? So 10 to the 3 grams, for example. You can put here kg, okay?
yeah.
But just for curiosity, you do this and you get 10 to the minus 12 grams. This is the weight of a bacteria, which is called, this is pico, 1 picogram. The mass of a bacteria is 1 picogram. Okay, you follow me? Very nice.
But when you are writing, we cannot continue writing with you.
I go fast?
Yeah, we will lose. If we could have your notes, it will become.
Well, this will be in YouTube, so you can, and then I pass you my notes as well.
It's hard to look at them again and start writing.
Writing will help you, I tell you. Even if you go fast, you put it here faster. It's good. It's good for you to write. Even if you cannot follow everything, it's good for you. I tell you, for me, as a learning technique, it's very good.
But when I'm writing, you say something that because I can't catch that, I can't understand the rest.
Okay. I'll try maybe to go slower.
No, I think having few notes with
Fine.
thank you.
I'll do this. Okay, this was about bacteria. Another important case is yeast. Yeast has a different structure, it's more spherical, and the size is a bit bigger than bacteria. This can be up to 10 microns, but it can be also smaller. So let's do it like this. And this one's the diameter. So let me call this the D. The diameter is of the order of 4 microns for the haploid. This has 1 copy of chromosomes, and 6 microns for diploid, which has 2 copies of chromosomes. And yeah, there's not much more we could say to highlight. Maybe that the cell wall, here, the cell wall is around 10 to 25% of the total mass. So, mainly what you have to remember is yeast is in general of the order or a little bit bigger than bacteria. And they're a bit more complicated in their function. They don't have flat gel, and they are very well studied in experiments. Finally, the human cells. As you saw in my presentation, there are many types of human cell. It's not the same a red blood cell than a neuron. It's not the same hearing cell, sensory cell. There are many types. So, human cells, I would say, is that there are around, okay, as I said, this could be one cell, but there could be also one disc, like the red blood cell, okay? RBC, HeLa, it could be the neuron as well, like this, very large, cell body, neuron. So don't think about the human cell as a fried egg, okay? It's not just like that. Okay? There's many types. Remember this, okay? And for example, the HeLa is 40 microns large. So this you have to remember, it's 10 times bigger than a bacteria. You have to think it's very logical. When we get a bacteria inside the cell, it's not that the bacteria is bigger than the cell. This makes no sense. It's smaller, also the virus. And yes, so important numbers, for example, is that there are around 3.7 plus minus 0.8 times 10 to the 13 cells in a human. So in our body we have 10 to the 13 cells, more or less. There are many. Another important thing is about the types. So there are around 200, exactly, 200 different cell types. So many. I put 3, but I don't know all of them. And the volume, so okay, the diameter D is of the order of 40 microns. But I put it here actually. And the volume is of the order of 500 to 4,000 microns cubed. Remember, the bacteria was 1 micron cube. This is much bigger. And of course, this is just an estimate, but what you think about in reality is that if you take a piece of skin and take it out and you measure the volume, as you can do now in the experiments, there won't be a number, there will be a distribution. This is very important for you to know. So there will be a distribution, like, it would be like this, for example. In the book of Miro you can find it for some experiments, and you can do here will be the frequency. So how many counts you get. You can do, for example, a histogram. Say, for this amount, I count, example, for 1,000. This will be the volume in femtoliters. It is important you know the size because of the order of magnitude. Because if you take the cells and you measure them, they will be of the order of femtoliters. If you do this histogram in meters, meter cubes, it will be nonsense. It will be 0.0000000. That's why it's very, very important in physics and in science. You know the units. The graphs they have much more sense. So, for example, I'm putting in the book they talk about the lymphoblast in mice. So there are some cells in the mice which have this distribution, and this can reach up to 3,000 femtoliters and start from 500 femtoliters. So you get, in many cases, broad distributions of volume. And yeah, with this, I give you already an idea about the first box. So let me go to the second one, which is about organelles. So up to here, I talked to you about this very briefly. Now I'll go in the second box, which is here. So we are doing like a zoom in a cell. We have the cell, not zoom when we are there, but the other one. We are in the cell, we're looking at cells, and now we go deeper, and we see the organelles. So we go to the organelles. I always forget the double L. Organelles, and the first one, okay, one organelle. What is the most important organelle for you?
Mitochondria.
Mitochondria is very important, yeah. There is one that has all the information about the rest.
DNA.
DNA, but DNA,
You can
the nucleus. So the first thing you see in a cell, the biggest organ is the nucleus of the cell. Well, now I draw it spherical, but I shouldn't. Okay, here's the cell, and then you have somewhere the nucleus. The nucleus. So the nucleus, it is around, for example, in yeast. Well, this one in yeast is one example. In yeast, the volume of the nucleus is of the order of 3 microns cubed. So it's a big piece of the cell, and it has 12 MVP. These are units for the genetic content. This is called mega base pairs. Mega base pairs. Base pair is a nucleotide. Sometimes you will see NT, sometimes you will see BP. This is 12 mega base pairs. Why base pair? Anyone has an idea?
Oh, because
The DNA, remember, it's like a ladder.
yeah.
A with T and C with T. Base pairs. So you have A with T, C with G, G with C. This is a base pair. A pair of bases. And this is 12 mega base pairs. Mega is 1 million. This is, well, there are 12 times 10 to the 6 base pairs. Okay. This is just to give you an idea of the language of the biologist as well, which is important.
We could gather all of the information in just a table and see that this is the table of the molecule and cell specifications.
Yeah, sure, it's good. But I'm telling you the story smoothly. The table is very boring, no?
Well, in this bit, I think it was better to go seeing the biology itself because we hadn't any lesson in the biology. We need to be more familiar with the cells, the structure of cells.
Yeah. No, the best thing in my experience is to have an image of the cell in the microscope. There's nothing better than that.
Is there a microscope here?
No, we don't have it.
No?
I wish, but this is the International Center for Theoretical Physics. So not only there's no biology, it's only theory.
Otherwise we could just make a human cheek cell slide.
Yeah, well, there are labs in Trieste, not in ICTP, but there are labs. This would be, yeah, maybe we could do something. I know some people who could show you.
Yeah.
Let me think about it,
You
okay?
can just swab your cheek and make a slide and look at human cells.
In my master for beginning work, my work with the microscope, my professor asked me to image onion cells.
Onions.
Yeah, I know that.
Wow.
It was really interesting.
Okay, we will think about it. Maybe when all your colleagues are here, we can go to a lab and see it. There are some labs we can see it. Let's wait for your colleagues. This would be better. All right. So and something else is like the volume. It's so the DNA inside the nucleus is not all DNA. The DNA is around the nucleus. It's around 3% of the cell volume. Right, and this is for yeast, but of course it's important to know what happens in the human cell. So let me just say something else about it. So of course, we always talk about the DNA, but in the end, it's not that the DNA is floating freely in the cell, but it's packed. This, I guess, you know well, no? This is in the chromosomes.
Yes.
So the chromosomes, it's like DNA, highly packed DNA, and okay, sorry. So the chromosomes in the IT are like structure like this. Okay. And here this is super packed DNA, in which if you zoom in, you will see, okay, again like a rock like this. And you zoom in here, you will see structure that looks like this. And finally you see discs, and it's not so simple as we thought. These are called some proteins that are called histones. And this is 8 parts. This is called the histone octamer. Imagine a disc with 8 parts. There are 4 parts and 4 parts, right? And around it you have the DNA wrapped. There's the DNA in loops here. This is the DNA. This is an important structure inside the nucleus. So the nucleus inside has the chromosomes. Yes. Like this X with this. These chromosomes, if you zoom in, you start to see very complicated structure. So it's not that the DNA is just floating, but it's highly packed in this with this protein. So this is called the, well, maybe this is not big enough, histone octamer, and this forms the higher level chromatin structure. So what happens is there are around 147 base pairs, and you can understand this, of DNA. DNA wrapped 1.75 times in these histones. It's not that you have one loop, you have one loop and 3/4. This is very, very interesting. The geometry in biology is very interesting. The radius of these histones, the radius is of the order of 15 nanometers, right? And the height is this, 8. 8 is of the order of 10 nanometers. It's a nano object, okay? It's not a microscopic, it's nanoscopic. And in human cells, altogether, make that the volume of the nucleus is around 0.2 the volume of the cell. This is also an important number. How big is the nucleus in comparison with the cell? 0.2, 30%, more or less. Its volume. We're not saying about the weight, we're saying about the volume. Alright? The next organelle I will discuss, this was about the nucleus. The next one will be the so-called ER. What is the ER? Anyone remembers? I'm giving a endoplasmic reticulum. The endoplasmic reticulum. This is very difficult for me to draw, so I will do my best. So imagine I cut the nucleus by half, and this is like the lower part of the nucleus, okay? The nucleus, and around it, there are like, well, I must tell you a very good story. Well, let me do the drawing and I tell you the story. And it reminds me, okay, and this is the ER. So the ER are like some membranes that are around, okay? The drawing is very bad, but they are like a set of membranes that are covering the nucleus. Another one here, another one here. It's like a maze of membranes. This is called the ER, endoplasmic reticulum. And it is very important to know. Okay, just to tell you one thing that is interesting is last year I gave an exercise to the students to make an analogy between the cell organs and different things. And one student said that these are the seats of the parliament. Okay. And I think this was excellent. In the parliament there are the seats. Okay? This is the plasmic reticulum. Very interesting, this exercise. So then one key point that you should know is that the ER is the largest organelle of all. It's the biggest, the largest in eukaryotes. Okay? In eukaryotes. In biology, you should know something very important. What biologists like is to know the structure and the function. You should know what is the function. So, you remember what is the function of a function? Mainly is processing proteins and transport. Its main function is to process. They process around 30% of the proteins, all the proteins, also lipids, and they store calcium. Like storage and also delivery. It's like a mail room, okay? You have the mail. This is more or less the idea. Another important, very important point is how big is these membranes that are here. This is a system of membranes, how big they are, and this is very spectacular because the surface area of this endoplasmic reticulum is around 10 to 20 times the surface area of the cell. You have the cell, this has a certain surface, and the endoplasmic reticulum has 10 to 20 times the surface of the cell. Okay? Much bigger. So we have something very, very wide wrapped inside the cell. I can put this page and I put this inside this bag. The bag is smaller, but I can wrap it very much and put it here. This is the idea. Okay? It's very important organelle. And okay, let me give you numbers of this. And now I'll give you what Said was asking, which is a table. So at least I will give you a table for the ER. But of course, it would be a nice exercise to wrap everything into a table, which is nice.
We need these numbers for simulations.
Yes, exactly.
So
Simulations is important.
each time we need these numbers, we have to reference.
Yeah, but actually I have many collaborations with biologists, and every experiment has different numbers. You can take the number that comes in a book, but this is for one type. Then you collaborate with some people, and they tell you, I work with other cells, not the ones in this book. So you have to ask them. So every problem in biology has a different size. This, what I'm giving you, is orders of magnitude. This is the only thing. So when you want to do competitive science, you need to know exact numbers. This is another story. We will go into this. Just one thing that could be interesting is how much is this in one example. So an example is the liver hepatocyte. These are cells that are in the liver. Hepatocyte. In humans. And you can build a table, which is the membrane type. And this. And on the other side, you put the percentage. Membrane percentage in the cell. So you have one cell, and you count the surface of all the membranes. And each one has a different percentage. One is the plasma. The plasma membrane is what you usually call membrane. So it's the membrane outside of the cell. Okay? That's the plasma membrane. This is just 2% of the membranes. The ER, to give you an idea, in these cells is the 50% of the membranes. So out of all the membranes present in the cell, in a liver hepatocyte, 50% is endoplasmic reticulum, from which 35 is the so-called rough endoplasmic reticulum, and 15 is the so-called smooth. But these are, of course, estimates. And there's more membranes. What else do we have with membranes in the cell?
The mitochondria has
The mitochondria. The mitochondria has membranes, exactly. The mitochondria has a lot of membrane. It's 40%. It's fun. But we have more. Repeat. Golgi apparatus. This is another organelle. This has 7% of the membranes. And the rest.
Doesn't the nucleus itself have a membrane as well?
Yes, but it's negligible compared to this. And finally, vesicles.
Yes.
And vesicles, like endosomes, for example. Endosomes. These are less than 2%. Just to let you know that these two are really big compared to the plasma membrane in the cell. Okay? Just to give an idea. And given that these two appear in mitochondria, so I think it's a good moment to talk to you about the mitochondria. So, I don't know if I have space here. So we talk now about mitochondria. So why mitochondria are important? Do you remember? What do we do? Something. Mitochondria. Exactly.
The solution can't have to be diluted.
It's energy. They produce energy. Very important for the cell. Yeah, shape. Okay, so I'm drawing on the top. This is the mitochondria. Okay, and inside, so here there's a membrane. There are membranes here. Membrane. This inner part is called the matrix, intracellular matrix, and the size is around 1 micron, and the width is around 0.5 microns. Okay. The function, as I say, is like an energy factory in the cells, in eukaryotic cells. They have size of microns, and it doesn't remind you this to anything we saw already.
The endoplasmic reticulum.
Well, a little bit endoplasmic reticulum because they have this type of membranes, but the size is 1 micron.
What was it?
1 micron, so it's the same size as a bacteria.
Yeah, and we actually think that, our cells captured it. So.
Exactly, so it is thought that the origin of mitochondria, one hypothesis is that it was a bacteria that was internalized or engulfed by a cell and then entered into a symbiosis with the cell.
Which soil are eyes?
Huh?
The eyes?
The eyes?
Are eyes, yeah. They're also thought to be like other organisms.
The eyes?
Yeah, eyes.
Well, I'm running this new now. The eyes come from another organism. We have to investigate this.
Yeah.
So the origin, there's a theory about the origin. Theory that came from endocytosis or endocytosis of a prokaryote. Which could be a bacteria. Bacteria by a primitive eukaryotic cell. And then they enter into a peaceful coexistence and are serving as an energy supply in cells. An interesting number is how many do we have? How many? So in yeast, there are around 10. But in mammalians, like humans, there are much more. There are between 10 to the 3 and 10 to the 4 per cell. There are quite so many inside the human cell. Right? Then there is a related organelle that is only present in plants, which are called the chloroplast. So I'll give you a little bit of numbers about chloroplast. It's only present in plant cells. This is what you should remember about it. So the chloroplast, the main characteristic is the color, green. Chloroplasts, these are different shape, very green, and they have different system of membranes inside. They have like stacks of membranes like this. Lipids by layers, and then we can produce sugar. And it is important. The size is around. This is around 1 micron, so they are bigger than the mitochondria. Again, the origin is similar. It is thought as well that came from free-living cyanobacteria that were engulfed billions of years ago. So it's similar origin. About the sizes, is the diameter is of the order of 4 to 6 microns, with a mean volume of around 20 microns cubed. And the structure, as I said, they have stacks of membranes. Stacks of membranes. And there's many important proteins inside, and one that is very important proteins. One is key, it's called Ribosome, among others. This one is very important in photosynthesis and in carbon fixation. Carbon fixation. It's a protein that is responsible for taking CO2 from the atmosphere and releasing O2 and keeping the carbon. So, very important nowadays. And I'll say a little bit about, again, how many do we have? Not we, but the plants, because we don't have chloroplasts. So, in algae, they're around 1%. But in other cells, like leaf cells, around 10%. Not so many as mitochondria. With this, I'm done about the organelles. So the class should last half an hour more, no? Till 3:30.
Yes.
Okay, so if you don't mind, I will continue because I have something very exciting to tell you at the end. So I prefer to spend more time with what I have at the end. So I'll pass this a bit faster. Okay. So I talked to you about the organelles until now, and now, as I said, I'm zooming in the cell, so I get into smaller and smaller and smaller pieces. First, I talked to you about cells, then about organelles, and now I'll go to the molecules, which is the smallest thing. Biomolecules. These are very important for cellular function, which is called in this book nuts and bolts.
I have a question about that.
Yes.
So in the notes that you gave us, you had written percentages, that 70% water, 26% macromolecules, and then 1% sugars, and all of that. But aren't the macromolecules made of sugars, lipids, proteins, amino acids? Like proteins are made of amino acids. So why are there 1% of the rest?
Why? Okay.
Are they like freely roaming in the...
No. So for example, you can have a sugar freely, you can have pure glucose, which is a molecule, yes, a molecule, but you can have sugars stack each other. You can have a polysaccharide molecule. So you can have a series of glucose one after the other.
Yeah.
Then you can have the molecules freely, but you can also have the molecules stacked into a bigger structure. Okay, so for example, the membrane is made of lipids, and it's one after the other. So it's not that the lipids are free. The lipids are one stuck to each other. So typically in biology, you are in a cell, and you don't have to think the cell as, this is very important, you don't have to think the cell as there is a nucleus here, and there is another guy here, and then there is mitochondria, and then, no. Things are very highly packed. Everything is touching everyone. So it's really, really, really dense. I can show you a video one day. So molecules are never free. It's not that there is a pool of water and there is a molecule here doing like this. No. You have to think about this as a very crowded room. Very, very, very crowded, where everyone is interacting with everyone. That's a better picture of this. But yeah, maybe there's glucose here. But then there will be here a polysaccharide, like many together. And then here, if you zoom in, you will see the lipids, and the lipids will be highly packed. So I'll try to give you a little bit about this in this part of the lecture. Alright, so the first biomolecule, so this will be biomolecule. The basic biomolecule, the first one I can think of is water. So water is extremely important for life. And way you can draw water, it's this. Now this will come to your question, you will see. Okay, this is water. Okay, H2O. You think about it like this, no? Fine. 2, 3 balls, 2 sticks. No, you don't have to think water about this. Water is a very dynamical object. Which actually it is charged. This is not a neutral molecule, it has charge. It means that here there is an excess of negative charge, and here there is an excess of positive charge. What do I mean by this? It means that the electrons that are sharing here, because this is a bond, so it means there are 2 electrons shared between H and O, covalent bond. The electrons are closer to oxygen than to hydrogen, because oxygen is heavier. Here it's the same. The electrons are closer here. This makes that this molecule gets a small extra charge, delta 2 delta minus, which is compensated by this delta. Well, compensated. Here they are charged. Locally they are charged. This is extremely important for life, because water is not that it's floating around alone. It's also close to other water molecules. And this is extremely important for life because there are networks of water molecules. They make a network tight together. Not tight, they are bound. For instance, if you have another water molecule here, and this, remember these are like 2 sticks that can rotate in 3D. It's not, it's in a plane. This is very important. Imagine you have now another water molecule like this. So you will have here the positive dipolar moment, and here the negative. So what is going to happen between this and this?
Attraction.
They will attract each other. Plus with minus attract each other. So what happens is that there is the formation of a bond between these 2. This helps water to be,
Hydrophilic.
this is called the hydrogen bond.
Yeah.
And it is formed throughout the DNA. In the DNA, when you have the A with T bound, this is what you have. It's called the hydrogen bond. Okay. This is quite important to know. So the hydrogen bond, this has a characteristic size. So this size is of the order of 1 angstrom. Remember, 1 angstrom is 10 to the minus 10 meters. This is 0.1 nanometer size. But the hydrogen bond is bigger, so you can reach longer distances with this. This is around 2.7 angstroms. It's a bit bigger. The hydrogen bond. And this bond is stronger. So it depends on how is the water. If the water is here, the bond is weaker. So it's stronger when these 3 atoms lie in a line.
Yeah.
This is the strongest configuration. Remember, this forms like a tetrahedron. Also this one. So when you put them like this, it's the strongest bond between the water molecules. And I think I told you a lot about this, but to me the most important thing is that you keep the idea that hydrogen bonds are very important too, because they appear in DNA. These are in DNA. When you have the ladder in DNA, like this, you have A and you have T. here in between, there is an hydrogen bond. Okay, and this with all the junctions between base pairs. Okay, also C with G. In the middle, what makes DNA stable is the fact that there are hydrogen bonds. This bond is of the order of the strength of the hydrogen bond is of the order of 1/20 the one of the covalent bond. Covalent. So it's not a very strong bond. Okay. Covalent bond is stronger than this one. This means that water is a dynamical structure. So there is a network of waters that are all the time breaking and making new hydrogen bonds. This if you think about it.
Covalent bond is for?
Covalent, the strength
Yeah.
of this bond is 20 times larger than this one.
Yeah.
It means that if you have these two molecules together, you will see 20 more times breaking of this bond than this one. Okay? And this is something I wanted to highlight to you. See that the importance of water for the structure, and the fact that it's a dynamical, it makes networks, it has fluctuations. So I'm studying a little bit water in doing research on water. So I'm learning a lot about its importance recently. And there's a lot of people investigating water nowadays.
Sir,
Yes.
I have a question. Since in this case we have two molecules of water,
Mm-hmm.
since the number of free electrons in oxygen are 0,
Uh-huh.
the same thing for hydrogen.
Uh-huh.
Then what's the meaning of hydrogen bond? Because you said you have this particle, this molecule and this molecule.
Yeah, it means that this one has 2 extra electrons and this one has 0 extra electrons. It's missing 1.
Which one?
H is missing 1 electron. So these 2 electrons that are here sometimes are passing like this.
Since we have hydrogen,
Uh-huh.
we have 2 pair of electrons.
Yes, yes.
But for hydrogen,
But-
it's
But one is doing a bond here and the other is floating around here. But the electrons are slightly, as I show here, are slightly displaced like this. So this has a net positive charge now.
net with one.
Net.
One.
Yes, net plus 1. So it's missing 1 electron.
Yes.
It's not that it's missing, it's that it's far from the hydrogen. So if you bring someone who brings an electron, and you are hungry for an electron, you will take it. Okay?
They're apples.
Huh?
He likes apples.
The problem is
What do you like?
He likes apples.
What?
Apples.
Apples. You miss an apple, no? And then someone passes with an apple near you, you will get closer to him, no? Okay? Then this is what is happening. It's getting closer. Okay?
yeah.
Okay, I'll give you the reference of a chapter where you can read this very carefully. The main point is that these electrons are getting slightly closer to O. They are shared between both, but they are slightly closer to O than H. So I'm sharing with you, but you keep the apple more times than me. So it's a bit fun. And then someone comes from here with apples. So, it's better I get closer to him, no?
Yeah.
This is what it is. Okay?
Okay.
Okay. So it's a matter of imbalance of charges. I'll give you the chapter where you can read this, but this is basically the idea. There is an asymmetric distribution of charges. The charges, there is here a bond, but the bond is a bit asymmetric. So the cloud of electrons are closer here than here. And this generates in H and net positive charge, small positive charge. So when someone comes here with a minus, you want to come with him. And this is what this means. Okay? There's an electron sometimes passing by here. This is my point. Okay? And, okay, in the end, this is a very interesting topic. In the end, when you have the O, the H, H. And then you have here an H. And then here an O. Here an H. Okay, you will have this bond here. Well, this bond is weak. It's not that it's made forever. It's like ceramic. It will be done and out very fast. But then this one will make another one here. It can make another one. So here it will be an O. Here with H. Here with H. So in the end you can build a network with water. That's what makes water to have this high surface tension. You can put a, you've seen in images, an insect walking on water.
Yeah.
This is because there is something holding, so there are some forces, and it's not only the covalent bond, it's also the hydrogen bond playing an important role. Anyway, I give you the reference you will see in more detail, because otherwise I cannot finish what I want to tell you, which is quite a lot. But okay, let me just be brief, at least with biomolecules. So this was the water, and briefly I comment on the others. Another one are sugars. Sugars are the main storage of energy. One is like this. This is called glucose. Here for details, you can check in the book about the structure. Here there are a lot of carbons, hydrogens and oxygens. So glucose has this chemical formula. Has the structure of an hexagon. And it has a mass of 180 dalton, which you can calculate directly from here. The total, okay, here there are carbons, and these carbons, this length here is 0.15 nanometers, which makes that the total length of the glucose is 6 times this. Nanometers, which is around 1 nanometer. So glucose is like a ring, and the ring has the length, total length of 1 nanometer. As I said, these are very important for energy. Another one, important biomolecule, are lipids. Lipids are key to make membranes. What do you say?
Fats.
What?
Fat.
Fat, exactly, fat. So it's like long-term energy storage, and the typical structure is written like this. Okay, this means that typically this is a COOH group, and here there is a chain of, you take these two, it's like C2, what is like, it's a hydrocarbon chain. So there is C, H, H, there's another C with H, and it's folded like this. What is important is that this has like a head and a tail. The head is hydrophilic and the tail is hydrophobic. The head is charged, and this is polar and this is non-polar. This is called the hydrophobic tail, and this is the hydrophilic head. What does it mean, hydrophilic and hydrophobic?
Hydrophobic is what repels water,
Repels
doesn't
water.
let it pass through. Hydrophobic.
And this likes water because it's charged. And this is, you see, I was explaining before. And this is non-charged,
It's also because there's no oxygen.
this is polar, and this is non-polar. The lipids, what is very nice is that they are very useful to build membranes. And in fact, cell membrane, let's say this is the cell membrane. Okay. This is the membrane of the cell, and here is the cytoplasm. Both out of the cell and inside the cell are filled with water. So if you want to build a coat for the water, you need something that repels water out of you. Okay. So the way this is done is that there are some molecules here that are these lipids, in particular phospholipids, which are arranged like this. So if I zoom in, what I see is something like this. These are the phospholipids. And this is the inner, this is the membrane. And out of them is the water. So, as I said, this is the polar head. This one likes the water. So these heads here like the water. And the other part, the hydrocarbons and non-polar, doesn't like the water. So water is passing here. Right? This is the way you build a membrane that protects the cell from the outside. This is the cytoplasm, and this is the inner cell. So I'm looking at here. Okay? Side, you're not convinced?
I couldn't understand this in the right. The
Okay.
membrane, the big picture of
Okay,
the
okay.
membrane.
So the membrane, let me draw the boundaries of the membrane in yellow, like this. Okay, if I zoom in, this is the yellow thing. This yellow thing doesn't exist. These are lines that I'm drawing to help you to understand. If I zoom in, what I see is this is a structure of a bilayer of phospholipids. So, in other words, here the heads like this and like this. Okay? And then they are like this. Okay, but this is very small. I'm zooming in. So they are arranged in bilayers.
Do you trace to each other?
No, they are giving the back to each other because these are the heads.
Yeah, heads.
Back to back. They are back to back. The heads look to water and the tail looks to the other friends. Okay? They are like this. This is called the lipid bilayer. Lipid bilayer. And what is interesting is what is the size of this bilayer. So this size here is 4 nanometers. That's a lot. 4 nanometers. So the membrane thickness is 4 nanometers. And if you compare with the cell diameter, the diameter of the cell is around 2 microns. Okay, 2 microns are 2000 nanometers. Okay, so you do 4 divided by 2000. This is like 1 divided by 500. So it's the thickness of this membrane is 1 divided by 500 the entire surface of the cell. It's very thin compared to the size of the cell. The thickness of the membrane divided by the cell diameter is 1 over 500. This you can compare with if you look at an airplane and you look at the thickness of the airplane, okay, round, you measure the diameter, that's 5 meters, its length, diameter is 5 meters, and the fuselage is the thickness of the paint outside of the plane. That's 1 centimeter, and this is again 1/500. Okay. To give you an idea of how thin is the membrane of the cell, it's like comparing the layer of paint in an airplane with its full diameter. Okay, this is to tell you it is very thin compared to the cell size. And it's 10 minutes. There's something I will skip. I will tell you the next day, because I have something very nice to show you in the presentation. But the last thing I'll talk about it a bit fast will be filaments. I didn't tell about the machines like the ribosomes and the RNA polymerases, their sizes, but I talk about it next lecture at the beginning, because I already talked about them, so let me talk about something new for you, which are the filaments. Filaments are other important structures which I didn't talk about. Filaments. I talk about, sorry, if you have told about this, because now I'm talking about the membrane, so talk about the organelles, talk about the molecules, talk about the membrane, the nucleus. But it's not only these spherical structures or volumes that are in cells. It's not only volumes. We also have ropes. We have filaments, which are important in the cell for cell division mainly, and for motion. One that I draw like this is actin filaments. I draw it as line, but they have a structure. And another that I draw. Hello?
This color doesn't show what in there.
Ah, that's a word. Okay. Do you see it like this? Do you see it now?
No, the problem they can't see the word.
Okay, this is fine. Actin filaments. Filaments. These are of the size, so they have monomers, and each monomer is of the size of 5 nanometers. They are quite long, so they can be of have thousands of monomers. And another one is very different are the microtubules. These are the ones I show you in cell division, when the cell was dividing. They are tubes that move the chromosomes. These are like hollow tubes, like this, and they have a very beautiful geometry, because there are filaments that are going like this. So there are several parallel filaments. And, okay, I'm not so good drawing, but just to tell you that these are called the microtubules. And they are hollow tubes. The step size here is 10 nanometers for a monomer. Remember, these are polymers, and they are made of repetitions of the same unit, which are called monomers. And the diameter is around 25 nanometers, and they are made of 13. Here we have something called 13 protofilaments. And I urge you to look at the reference, because I'm not able to draw this properly. Okay. And what is important is their size, length. Length is around 10 microns. So they are really big. They can take almost the size of the cell. And the persistent length, well, I cannot enter very much into this, but just to tell you a little bit about their function. So actin filaments, they form the so-called cytoskeleton, which is like, well, this skeleton is like the bones of the cell. They form fibers that are all around the cell. Actually, the muscles are made of this, actin and myosin. Actin are the fibers, and myosin are the motors that pull the fibers. And I'll give you models about this. Actin filaments are responsible of the motion of the cell. So these are polymers that are growing like this when the cell wants to move. The cell wants sliding. To slide, it has to grow a rope and move. So they push the cell to grow. They generate motion. The microtubules have another function, which is cell division. When you have a cell that divides, there are these structures called spindles. It's called spindle, which is made of microtubules. There are like two poles in the cell, which generate all the microtubules going like this. And in between, you have the chromosomes here, which then are pulled apart and get cell division. Okay, these are the chromosomes. So microtubules are involved in cell division. Cell division, but also transport. So on top of the microtubules, you can have a machine, a motor that is moving a load. You can have a machine that is moving a load on top of microtubule. These are called the molecular motors that I'll discuss very much during my course later. It's 3:25, and I wanted to show you something very exciting in the presentation that I brought today for you here. So I have to share screen with your colleagues.
And the projector.
Huh?
The projector.
And the projector, very important. The projector is on. So since we are in this pandemic, today I brought something about coronavirus. Not only because of it's important nowadays, also I'm doing some research on this topic with other collaborators. So I'm happy to share with you some of this research. So we'll go now into slides only. I will share this. So you tell me from.
We can't see.
You can see?
It's too dim.
We turn on the light.
Yes.
Please don't sleep. It's here the light. Like this? Okay, guys, do you see me? Do you see my presentation? From Idris, Anna?
Yes.
Yes.
Very nice. So I'll talk briefly about the coronavirus. So I was telling you the structure of the virus is a bilayer, like the one I'm explaining to you now. And these red color structures are proteins. This is called spike protein. And you see it has a form of, from outside, like a triangle. This triangle is because it's a trimer. There are three proteins that are equal, assembled in the form of a triangle. It's a trimer of spike protein. So this is from a microscope. This is a real image. It's not done with a computer. This is real. And here are different parts that you can find now in many articles. So you see the S is the called spike protein, glycoprotein. You have the bilayer, lipid bilayer, similar to what the one we said. Still, it's not known perfectly the structure of this layer. There are proteins. There is an envelope protein, and there is also RNA inside the virus. Right. which is also the size of nano. And I'm going to show you, this is one of the receptors that are involved in the infection of the human cell by the virus. So the virus is thought that it binds to the called ACE2 receptor. It's called angiotensin converting enzyme 2. This is a very complex process that we are still learning what is going on. You have a question? No, okay. So this is a simplification of the process in which we say the human cell recognizes the virus by binding. So the virus has like a key. It finds at the gate, goes to the key, and it's entering through this receptor ACE2. When the ACE2 feels this spike protein, it says, okay, you are something nice for me, and it opens the membrane to the entry of the virus. At the molecular scale, you see something that is on the right side. So on the right side is a structure you can find online. I downloaded this structure online. This is called Pdb File. Pdb File is a file of the structure of a protein. And nowadays, there's many researchers working on this. So you can find this structure online and download it. What I show in the red is what we are studying now in ICTP. We have a big collaboration for this. The blue thing on top is the receptor binding domain. So we are taking from the trimer only one of them, the monomer of the spike protein, and we look at, okay, we wanted to look at the binding with ACE2, which is the yellow, but what we wanted to do is to find a molecule that blocks this junction. Okay, so doing this in a computer is very complicated if you want to do a simulation. Really, really complicated. So we did something much simpler, which was just the top thing, the receptor binding domain, in water. So we also simulate the water molecules. And we try to find a molecule that binds to this binding domain in the same place where ACE2 is binding. This is something we investigated recently. And I'll finish showing you some very brief results. So we look at this molecule, it's called lecithin, which is a phospholipid. So it's similar to what I'm showing here. So it has a non-polar part and a polar part. And well, this actually you can find the article already. And this person who did the simulations was a diploma student 5 years ago. So then he did PhD. So this is for you to see. He was diploma student in CMSP, another program. And then we did a very good collaboration. And on the right you see the spike protein is this structure with many colors. These lecithin molecules that are the ones here. And all these are water molecules, the gray. So we look at simulations of this system, and just to show you, this is what they do. So they bind in many places, but the red one is the hot spot, is where the cell, where the virus binds with ACE2. And this is for low concentration, but if you put high concentration, this gets very nice.
Is that all water molecules?
Yeah, water are all around. Are all around.
So water can block the virus?
Yeah, water is important in blocking the virus. What we see?
This particular virus.
Yeah, well, these things that are attaching, you see, this is not water, this is lecithin, this molecule.
Okay.
And these values, they get stuck in the place where
it's supposed to.
to bind the human cell, and the binding is very strong. So they get there for a long time.
So this could be a potential
vaccine. This could be, but you can put other molecules that bind. I'm not saying it's the only one. We are investigating one molecule, but there are many. So this is a very open area of research, very active. So I just wanted you to see what many people is doing. We are doing this, but we don't do experiments, which is more relevant. But we are trying to help in this crisis, and there are nice topics to investigate. But just to give you an idea of molecular interactions and applications to virus. So I think we have any doubts, any questions? Watch the movie for a long time. It's very like, it's a GIF, so it enters a loop. This is a GIF, so it's a loop. It finished and then starts again. To give you an idea, this was done the simulation took months to do it. And the time we simulate is 1 microsecond. Only 1 microsecond. Okay? because there are many atoms. So it takes a long time for a computer to simulate this. There are many atoms. There are 40,000 water molecules, then there are 10 of these, and then there's the protein. See, making a simulation of this is very, very hard computationally. Very hard. Sorry.
How did you isolate just this receptor? One of the...
No, it's not that we are isolating. We are doing a very simple simulation where we don't have the rest of the world. Okay, so this is many times we do this in physics. We isolate one part. We want to understand a small piece of the problem with physics.
This is Python simulation.
No. This was done in Gromax, which is a software to do this type of simulations. It's not done in Python. No, in science, everyone has its own tool for different problems.
Yeah.
So Python is good for example, stochastic process, which I will explain you later in the lecture, which is not what I do. For this, my collaborators to use Gromax.
Because I wanted to know that Python has the libraries for showing pictures like this.
Yeah. This was done, the graphical representation with BMD. It's another program. It's open source, you can download, and but it's not Python, it's another language.
Yeah, I see.
Yeah, it's good you learn one language, but in the end you will discover there are many useful for different projects in science. Okay, last question because we are a bit...
Yeah. You use the stochastic process in this work?
Well, here it's Newton's law actually what we use. But there is a bit of stochastic process because we say that this is in a thermal bath. So we have to give a way to show that this is in contact with a thermal bath. This is used using random numbers. So there are stochastic process here. But let me go further in the course and you will see how stochastic process come. But you could do a stochastic process description of this. This is done using Newton's laws and some potentials. You let the system go. But it will come. Stochastic process is important. Any other questions? Okay. So I'll finish with this. Thanks for your attention and we'll meet next Tuesday. Thank you.
Thank you.